Good morning, students. Today we will see about the retina. Before seeing the retina, today the detailing we are going to see how the development of retina, what is the blood supply of retina, what all the layers are present in the retina. First, before going to see the retina, we'll see about the overview of the structure of eye. In last class, we have seen about there are different layers of eye. Outer layer is a protective layer, and inner layer will be the vascular layer, or else it is called the uveal path, and it is highly vascular, rich in blood supply. The inner layer is a neuronal layer. The inner layer consists of pigmented layer and the neuronal layer. This inner layer is called the retina. So the retina is the sandwiched between the vitreous humor and the choroid, right or not? This is the retina. If you cut this ora strata, where is ora strata? Is the this is a corneoscleral junction. In the corneoscleral junction, there is a finger-like projection will be present. It is called the ora strata. It is a rich number of rods and cones are present in this area. If you cut the small portion of this retinal layer, if you see in microscopic, you could see this is a sclera, this is a choroid, this is a neuronal layer. Neuronal layer is called the retina. So the retina is not highly vascular. It's a nerves, that is a photoreceptors are present. Rods and cones are present. Rod is a rod in shape. So rods are in a rod in shape. Rhodaxin, the pigments are present in the rods called rhodaxin. This is the structure of rod. This is the outer segment of rod. This is the inner segment of rod. This is a nucleus. This is a synaptic terminal. Cones will be in a cone in shape. Nearly we have 100 million rods are present. Cones are 5 to 7 million cones are present in retina. If you see the development of retina, during the 4th week of the embryonic development, embryonic means the fetal development, from the prosencephalon, there are two vesicles are coming out. From the two vesicles are telencephalon and the diencephalon. From the diencephalon, the outpouched, the cup-like a structure which is outpouched from the both the side, that is called the optic disc. So from this optic disc, the retina is being developed. Inner layer being developed, then neuronal inner layer is a pigmented layer. So this is the neural tube. There is the forebrain, midbrain and the hindbrain. So in the prosencephalon, the vesicles are started. In the inner part of the vesicle is called the diencephalon. In the diencephalon, there is a outer pouch, like a cup like a structure is coming out, where the retina is being developed. The blood supply of retina mostly it started from the ophthalmic artery. In the ophthalmic artery, the main branch is called the central ophthalmic artery. This central ophthalmic artery divides into, in the posterior side, it is called the posterior ciliary artery. Why we are mentioning here? Mostly the posterior ciliary artery is only major supplying the blood supply to the pigmented layer of the retina. There are many arteries in the branches. Ophthalmic artery, the central branch, it's entering through the optic tract, and it is supplying to the pigmented layer of the retina. Then since it is a posterior side, posterior short ophthalmic artery, and then ciliary artery, and short posterior short ciliary artery, and then anterior ciliary artery, which is supplying in the anterior side of the ciliary body, iris, everywhere this is supplying. Now we are seeing this artery mainly this is sclera. When we see the layer, how this posterior part, this is the one, this is artery in an enlarged manner, it has been shown. It is the one mainly, it is supplying the blood supply to the pigmented layer of the retina. It is mainly supplying the blood supply to this main area. If you see the cells of the retina, there are 4 major cells are present. So that's the epithelial cells, and the epithelial cells, and then epithelial cells and photoreceptor cells and neuronal cells and glial cells. All these cells are present. These cells are arranged in order. These arranged order only it's creating retina. So in microscopically there are four kinds of the cells which is making the retina. That's the epithelial cells, photoreceptor cells, the neuronal cells, and the glial cells. We will see these cells only arranged in a systematic order, and they are forming the layers of retina. We'll see how these retinal layers are arranged. This is a sclera. This, as I told you, this is a posterior ciliary artery. These are all the capillaries. These are the pigmented cells. It is tightly been arranged. So it's present melanosome. It is present. So this is the first layer. We can say this is the first layer of the retina. Since it is a pigmented layer, we can call it the pigmented layer of the retina. The next, in this pigmented layer, they are getting nutrients from the posterior ciliary artery capillaries. It is supplying the blood supply. Okay, and the next in the this is the outer side of the retina. Outer side of the retina. This is the inner side of the retina. So you should not confuse it. In the pigmented layer, rods and cones are arranged. We have already seen how the rods are there. Rods are in a rod in shape and cones are in the cone in shape. Rods are 100 million rods are present, and the cones are only 5 to 7 million cones. This is the synaptic terminals and this nucleus, and then rods, the pigment are present is called rhodaxin. So this layer, all the neuronal layer, photoreceptor layer, photoreceptor sensitive area, this is very much. And if you see the next one, these terminals are connected. If you see this violet line, these 2-3 rods are connected together, and it has got a nucleus, and it is connecting with another one that is called glial cells. G is called the glial cells. So this both the sides are connecting, so it is called the bipolar cells. Bipolar. So these bipolar connecting with the glial cells. So the glial cells continues with the nerve fiber, and nerve fiber connected with the foot plate. This is the one. So now this is one string come. If you closely see, many rods are connected together, but the cones or the single line will be coming. Cones will be connected in a single form, not by this one. So very less in number. So this one. Then this one you can remember as RBG. Okay, RBG. It is a easy form you can remember. Okay, for receptor cell, bipolar cells, and glial cells. This we have seen. Now we will remember another mnemonic is called the high cells. So high cells is nothing but the horizontal cells. So these are all H is this HH. This is both are connected. Is called the horizontal cells. It is like a circuit. Retina is like a small electrical circuit. This is called the high. It is in a reverse form. In this connecting is called this amigron cell. So it is called the A. It is a it is in a reverse side which is connecting is called the amigron cells. So okay, amigron cells. And one more thing, important thing you have to see. All these cells are connecting together. Here they are forming like a network, correct? So this network, it looks like in a microscope. If you see, it looks like a mesh. So it is like a flexiform in structure. So it is called it's the outer side of retina, right? So it is called the outer flexiform layer. Okay, here one forming outer flexiform layer. Same way, here many cells are connected together. Here this structure together, it's forming. This is called the inner flexiform layer. This both outer flexiform layer and the inner flexiform layer are connected by inner connecting membrane. So it is called inner membrane it is connecting. So this is the interflexiform cells. This because it's connecting both this one both flexiform layer, outer and inner. So it is outer layer, outer flexiform layer, inter. So it is called the interflexiform cells. So these interflexiform cells takes the information in reverse direction. So outer to inner. Understand? So these high cells are H and all, these are all called the horizontal cells, and amigron cells also taking the information in a lateral way. High cells and amigron cells is like a horizontal, in a lateral way it is taking. So bipolar cells are also so when RBG cells takes the information from RBG cells takes the information from outside to inside. When the light falls into the rods and cones, they produce some graded potential. So these graded potential they modify the activity to the bipolar cells. The bipolar cells both the sides are connected, right? So bipolar cells also generated the granted potential, and next bipolar cells influence the impulses generating to the ganglionic cells. So the ganglionic cells also produce the action potential. So these ganglionic cells, these action potential or goes into the optic disc of the eye. So optic disc of the eye, it goes to the central nervous system. So the optic disc takes the information to the central nervous system. So the flow of signals of the retina is always from the outer side to the inner side. So according to this one, so where the light is coming from the RBG cells to that each cells are taking. And one more important thing we have to see is called the black color structure is called the mom cells, or otherwise called the Muller cells. So these mom cells mainly it is connecting, it is holding the retina, and other cells are in a tightly in its own place. Otherwise what will happen, it will short circuit. So it holds in the place. So it will holds in the place and it is continuing. It also have a nucleus, and it is extending up to this one and to this till the foot plate. Understand? So we have seen this is called sclera. This is the posterior ciliary artery, capillaries of posterior ciliary artery supplying the layers of retina. So now what we have seen, this is called pigmented epithelium. Rods and cones are present. Rods and cones terminals are connected by bipolar cells. The bipolar cells are connected with the ganglionic cells, and ganglionic cells continues with the neuronal fiber, and it is entering. The fiber entering into the optic disc. Okay. Then only those cells are there, that is called RBG. Then next will be high cells, and then high cells are nothing but it's a horizontal cells. Okay, these are interconnecting will be this horizontal cells, and reversely it is connected is called the amigron cells. And all these connecting place, it's called like a network, like a junction, it's called the plexiform. Since it is outside the retina, it is called the outer plexiform layer, and the this one is called the inner plexiform layer. When you see in the microscope, it looks like a small layer, right or not? And these two plexiform layers are connected, it's called the interplexiform cells connected by this. Understand? So now after drawing this picture, if you want, clearly you can see in this picture, this is the how. Okay, what is this? This is a choroid, capillaries are supplying. This is all the epithelial layer. So it is called the pigmented. This is the first layer. So it is called the pigmented layer. Okay, pigmented layer. This is the first layer of the retina. Next is the layer of rods and cones, because the rods and cones are arranged in order. So it's called the layer of rods and cones. And if you see in microscope, this mom cells nucleus forming, that is one membrane, black color membrane, like all these in the microscope it is showing. So it is called the outer limiting membrane. So it is called the outer limiting membrane. Next you could see the nucleus of rods and cones. So it is a nuclear layer of rods and cones. Then will be the outer flexiform layer, and will be the nucleus of the bipolar cells and mom cells. So inner nuclear layer, and this one is called the outer flexiform layer. Then will be the eighth one will be the ganglionic layer, and the neuronal nerve layer. Neuronal layer, you can say nerve fibers are conducting, so nerve fiber layer, so it's called the neuronal layer. And this one is called the inner limiting membrane. Remember, it is not outer limiting membrane, it is the inner limiting membrane, because light is transmitted from the outside to the inside. Are you clear about it? So these are the different layers of retina. So in this class we have seen how the retina been developed in the 4th week of the embryonic life. You know very well from ectoderm, like various changes like coming in this neural tube is forming, and neural tubes are bulging and forming the like a brain, right? Forebrain, midbrain, hindbrain, and hindbrain are developing laterally. Ventricles in the cavities forms, and ventricles, cerebellum, all these things started to develop. So various cells, it is evolving from that, right or not? Prosencephalon. If you take the forebrain, prosencephalon are some vesicles are forming, and in the telencephalon and the diencephalon. So in that only, in the down, the retina is coming. It's called the optic disc, where that pigmental layer started to develop and blood supply started to develop, and the inner will be neuronal layer. So this is the development in the 4th week of embryonic life of the retina being developed. Then you could choose the blood supply. Always blood supply, you can remember the ophthalmic artery when it is supplying to the central, and then when it is divided into inferior, superior, you can tell, and then another artery will be the posterior celiac artery. In this is long arteries there, short arteries there. When it is supplying to the front side of the eyeball is called the anterior ciliary artery. So this is there are but main branches coming in the ophthalmic artery. So if you see posterior ciliary artery, why we are mentioning here, these capillaries only mainly supplying the layers of retina. So what all the layers are there, immediately you can remember about RBG and high and mom cells or Muller cells, right or not? So there are various kinds of the cells which are present, right? The epithelial cells, like neuronal cells, glial cells, so pigmented cells. So these cells are forming the retina. So according to this arrangements, this is outer side of the retina, this is the inner side. Light waves are coming and passing, and then it is changing into the graded potential and going, and then it converted into the new nerve impulses, and it is going to the optic disc, and it is going to the central nervous system. So according to this, now we are focusing on the layers of retina. So the first one is called this pigmented layer. So the pigmented layer of the retina. The second one is the layers of rods and cones, and third one the outer limiting membrane. This is nothing but the mom cells which is forming. So it's holding the rods and cones in its position. So it, when it look in microscope, it looks like a membrane. Since it is the outside of the retina, it is called the outer limiting membrane. Then all the nucleus of the rods and cones. So the nuclear layer of rods and cones. Then this network is together is called, looks like a one layer of retina, that is called the outer plexiform layer or the inner plexiform layer. Understand? And why it is forming? Because high cells are there, horizontal cells, amigron cells. So interflexiform cells also connected reversely, it is called, taking the information in reverse, right or not? And then another 6th layer is called the nucleus. Like what nucleus and all is present? Bipolar cell nucleus is present, and the Muller cell nucleus, all nucleus forms like one layer. So it is called the inner nuclear layer. Okay, that is the 6th layer of retina. Then next if you come, that's another flexiform, that's the inner flexiform layer. Since it is outer, this is called the inner flexiform layer. Then this one is called glial cells. So there's a glial cell layer of retina, glial cell layer of retina. Then from the glial cells, the nerve fiber. So the neuronal fiber layer of retina. Then will be the inner limiting membrane where the Muller cells, where it is holding, that's called the outer limiting membrane. This is called the inner limiting membrane. Understand? So this is the structure of structure of retina. This is the layers, different layers of retina. So overall, the retina is the important retinal layer. It is the innermost layer of the eye. It is sandwiched between the vitreous humor and in the choroid. It is taking its own blood supply and nutrients from the choroid and also from the ciliary artery, that posterior ciliary artery. And if you see the cut section of this retina, there are 10 layers. There are various cells forms the retina: epithelial cells, glial cells, neuronal cells, pigmented cells. Many cells are forming the retina. Retina is mostly developed from the fourth week of our embryonic life in the diencephalon of the forebrain. Diencephalon, it is coming, it is like from the prosencephalon where the it is developed main blood supply. So if you write this much in nursing is more than enough. You will get 5 out of 5 or 10 mark, means 10 out of 10 mark. Surely you will get it.